Hello everyone. In this second video of the chapter on gene expression, we are going to address translation, the process of translation. We are going to address the genetic code. Then we're going to look at protein structure and the folding, how it happens, and finally some issues when proteins are misfolded. So what is translation? Translation is basically the conversion of the nucleotide sequence in the messenger RNA into an amino acid sequence. That's translation, right? So proteins are going to be built from the information that is contained in the messenger RNA sequence. A particular codon in the messenger RNA is going to have a corresponding amino acid. I addressed this earlier when we talked about the transfer RNA, right? We did say that a transfer RNA, it's like in form of L, like this, with a part here where an amino acid is carried and an anticodon here that recognizes the codon that is carried by the messenger RNA. The organelle in the cell that does translation, it's the ribosome. So the ribosome is responsible for recognizing the codons on a messenger RNA. Notice that the codons are recognized 3 nucleotides at a time. And so that's 1. 2, it's that as the codons are recognized 3 nucleotides at a time, which makes it actually what we call a triplate. Triplate. Triplate means 3, right? So 1 nucleotide, 2 nucleotides, 3 nucleotides. And another triplate, it's 1, 2, 3. And another one, 1, 2, 3. Now, notice that each codon on this messenger RNA is recognized 1 at a time. There is no overlapping. So first, there is codon number 1 that is recognized, and we identify the equivalent amino acid. So AAG, the equivalent amino acid is lysine. Then the ribosome is going to read UCA, that's the second codon. So codon number 2, the equivalent in amino acid for that codon is serine. And then you have GUC, the codon number 3, the equivalent amino acid is valine. Now, notice that as the ribosome reads the codons in the messenger RNA, they are actually going to connect amino acid to each other via peptide bonds. So this is a peptide bond. Peptide bond. So that we are going to have a polypeptide. All right? So translation, it's going from the information in forms of nucleic acid in the messenger RNA to information in terms of amino acids that are attached to each other in a polypeptide. All right? Now, obviously, if you go back in gene expression and specifically in transcription, you will know and remember that we did say that the DNA that is a double-stranded molecule for a given gene, you are going to have a coding strand and a non-coding strand that is used as a template. That non-coding strand that is used as a template is this one, so that you can have the coding messenger. All right? Please remember that. Now, the next segment, it's now that we know this, what is the genetic code? The genetic code, it's basically how the cell reads the genetic information. And what we notice is that whether a cell, it's a bacterial cell, a protist, a eukaryotic cell, a skin cell, a liver cell, cells of a human, cells of a tree, like an oak tree, cells of a dog, they all read the genetic code the very same manner. I'm going to address that. That's actually what researchers found, right? So across all living things, the genetic code is the same. This is a tremendous information to have, because now we realize that, wait a minute, a plant, a human, a bacterium, right? They all share the same genetic code. That is tremendous. And let me jump ahead here and tell you that, for instance, when you consider human insulin, if you consider people who have diabetes and who are actually taking insulin shots, these insulin shots, human insulin, are not produced in a human being because it would not be ethical to do that, right? However, we can wonder, how do we produce human insulin? And we find it at CVS Pharmacy, we find it at Walgreens, at Drug Mart, and so on. How is that possible? Well, what we do is that we take a segment of the human gene for insulin, the open reading frame. Remember the open reading frame? I'm going to address that again a little later. The open reading frame only. We take that open reading frame of the human insulin, we insert it in a bacterium, and the bacterium, E. coli, is the one that we use in the industry to actually manufacture the very insulin you find at the CVS Pharmacy, at Walgreens, at the Walmart Pharmacy, and so on. Obviously, we purify that insulin so that it is safe for human to take, right? Think about that a human gene is expressed in a bacterium that is completely different from human, right? That speaks to how universal the genetic code is. But let me close that parenthesis. So the genetic code, it's basically the correspondence that is drawn between a chemical language of the messenger RNA and the amino acid language that forms proteins. Now, the work started long time ago, but really tremendous achievements were made in the 1960s. In the 1960s, researchers actually used logic and very smart experiment to show that the genetic code, it's basically a shift from from a nucleotide sequence to an amino acid sequence. But how do they show that? I'm going to give you some aspect of how they show that, but not everything, because it's kind of complex. Anyway, so what researchers found, it's that the nucleotide sequence in a messenger RNA is read 3 nucleotides at a time. Now, you remember that when we consider a messenger RNA, the nitrogenous bases are A, C, G, and U, right? Now, if the system is reading 3 nucleotides at a time, that means that we can have A, C, G. We can have C, G, U. We can have U, G, C, which is not the same as C, G, U. We can have C, U, A, and so on and so forth. And what the genetic code shows is that for every single of these 3 bases, you have an equivalent in amino acid. So here is how biologists actually find out what the equivalent in amino acid is. So say we have a sequence, that of ACG. How can we determine the equivalent in amino acid? So A, the first letter, it's right here in this column, right? The second letter is going to be on this line. On this line. And the third letter is going to be right here, 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 and here. All right? So now, considering this, let's determine the amino acid for ACG. So A, it's right here. So that's number 1. C, it's right here. That's number 2. G, now we are going to be A, C, and then we are, so A, it's right here. C, it's right here, right? And G, it's right here. So that G, we are going to come this way and find that my ACG, it's right here. And that ACG encode the amino acid threonine. All right? How about CGU? C, well, let me change the color so we can follow. All right. So C, it's right here. And the next one, it's G. Where is G? G is right here. Okay. So now C, where is C meeting G? It's going to be right in this rectangle. Now, CG, the last nucleotide is U, CGU. So CGU is right here. What is the amino acid for that? It's arginine. And we can do this for whatever sequence. Another one is UGC right here. So UGC, let's change the color. Okay, so UGC, so now we have U right here. And where is G? We have G right here. Okay, so we are going to be in this area. Okay, now finally, where is C? Oops, I'm sorry. Oh, come on. C is right here. So now we have UGC, and the amino acid is cysteine. All right, so this is how we read the genetic code. Okay, and again, the genetic code is 3 letters of nucleotide for 1 amino acid. All right, so let's move on. So we have a triplet code. So 3 nitrogenous bases, right, or nucleotides for 1 amino acid. And basically a triplet is a codon. Now, here is the thing. We have 4 nucleotides possible, right? So we have A, G, C, and U. The question we are going to have is this: what are the number of combinations possible between A, G, C, and U to have the 20 different amino acids that are found in nature? We know that if one single nucleotide was to represent an amino acid, we would only have actually 1 amino acid for A, 1 amino acid for G, 1 amino acid for C, 1 amino acid for U. Now, we know that in nature we have 20 different amino acids. Here we only have 4 for 1 nucleotide for 1 amino acid. Therefore, the genetic code cannot be 1 nucleotide for 1 amino acid. All right? So let's say this. Then the next possibility, it's that, let me change color here. The next possibility, it's that 2 nucleotides have an equivalent in amino acid. If we do that, then we say, okay, so 2 and 2 and 2 and 2. So you're going to run combinations for this 4. So actually then you're going to have 4 square. This is 4 times 4. This is 16 combination possible, right? Which means that we would only have 16 amino acids. But we know that in nature, we don't only have 16 amino acids, we actually have 20 amino acids. So we are short. Then the last option is going to be, okay, let's bump it up. Let's say that rather than 2 nucleotides for 1 amino acid, we are going to say 3 nucleotides for 1 amino acids. So our A, G, C, U is going to now be, so for a different color, please bear with me. So we're going to say AGU, it's a possibility. GCU, it's a possibility. Then we're going to have A, G, U. We're going to have G, A. It's a possibility, right? So you have many possibilities, right? But how many possibilities do we have? The possibilities we have is going to be 4 cubed, which is 4 times 4 times 4 possibilities. That's 64 different codon possible. Now, if you say we have 64 different codons possible, this implies that for the 20 amino acids that exist in nature, some of them actually are encoded by different codons. In other words, you're going to have one amino acid for multiple codons for some of the amino acids, right? That is actually what biologists discovered, okay? And they found that if you modify the sequence of nucleotide in such a way that you take away one nucleotide or two nucleotides, you are going to change the sequence of amino acid that you get at the end of the day. Okay, so first and foremost, biologists found that you have in a messenger RNA what we call a reading frame. And that reading frame is 3 nucleotides at a time. Okay? If you have a deletion of 1 nucleotide or 2 nucleotides, that's 1. And 2, if you have an addition of 1 nucleotide or 2 nucleotides, you are going to have what biologists called a frame shift mutation. Okay? That frame shift mutation means that you are just simply going to change actually the reading, right? Rather than, for instance, if you have A, C, G, A, U, G, C, C here. So you have ACG, AUG. Let me add another nucleotide to make it fair. And CCG. That's your frame, your reading frame. If you lose one of this nucleotide, then you're not going to have ACG anymore. You're going to have A, G, A, and then U, G, C, and then you're going to have a CG here that is going to have no equivalent in amino acid. If you lose a second nucleotide here, then your new frame is going to be A, U. Then you're going to have G, C, C, and G, right? So you're going to have a different set of amino acids, right? So that is a frame shift mutation, right? Whether you are actually losing some nucleotide or adding some nucleotide, you are going to experience a shift in the frame. All right? That is very important to remember. That's why mutations are very bad when it comes to integrity of the proteins that is produced when a gene is expressed. Okay? All right. So now, based on the fact that a codon is a triplete, right? If we start with an original sequence here that we're going to call wild type, that wild type sequence is going to be a succession of GAC, 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 the equivalent amino acid is going to be asparagine, 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 asparagine. Now, say we have an insertion of one nucleotide. That insertion happens in here. So now we have the addition of a G. Now, what was GAC? Instead of GAC here, now we're going to have GGA. And now everything else is going to be changed. As a consequence of that, let's take a look at the sequence of amino acid now, the new sequence. We have asparagine, glycine, arginine, 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 which sequence is completely different from what we start with. Let's see if we add one additional nucleotide to the first one. So we're going to add this time U in addition to G. Now, we still have our GAC to start with, but now we have UGG, which is tryptophan, and then we have ACG, 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 which is threonine. Okay, a complete different sequence. So you see that whenever you have an addition of one nucleotide and two nucleotides, we have a frame shift. Now, what if the mutation is such that you are adding 3 nucleotides? If you are adding 3 nucleotides, let's see what happens. So you add UUG. So we have GAC, UUG, it's equivalent in amino acid, it's leucine, and then after that you have GAC, 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 GAC. You have the same sequence, right? But with the addition of a leucine, which means that your sequence, while it is different from the original, you only have one amino acid that is added, everything else is the same. While in a shift frame, a frame shift, the sequence is completely different, right? That's why insertions and deletions are bad news, right? Please remember that. Okay, so what researchers were also able to show is that the genetic code is not overlapping. The genetic code is not overlapping. It's actually 3 nucleotides, one after another, right? Which means that this is going to make the reading much easier. If not, if the genetic was overlapping, this would have created a bunch of constraint in protein structure. I'm not going to take you into the details of that, but let me just mention that. And also, the open reading frame is going to, the system is going to control where the open reading frame starts and where it ends. Okay? So, in the messenger RNA, you have a number of nucleotide sequences on the messenger RNA that indicates the system where to start and where to end. Okay? And now, as we know where to start and where to end, in between, you are going to have the open reading frame. And when we consider that open reading frame, right, we don't consider the end part or the beginning part. These two parts are not part of the reading frame, what a protein is going to be, right? The beginning and the end are only there to guide the ribosomes to the right open reading frame. Very important to understand. So now, when we have a sequence, let's have an imaginary sequence. This imaginary sequence right here. AUGCC CAAGAC. If, say, we identify a new gene and we don't know the sequence of amino acid for that new gene, we are going to ask, okay, well, what are the possible reading frames in that sequence of DNA? That's the question we are going to ask. And when we ask that, we are going to say, okay, let's assume that the ribosome will start reading from A. So, if we start reading from A, then the sequence is going to be AUG, CCC, AAG, and this doesn't account for any amino acid. So, therefore, the first reading frame is going to read methionine, proline, lysine. All right? Then, let's shift. What is the second possibility of reading frame? The second possibility of reading frame, we are going to disregard this A, and we're going to start reading from U. So now we have UGC, then CCA, then AGA. This C doesn't account for any amino acid. With that, we have UGC is cysteine, CCA it's proline, AGA is arginine. That's the second possibility of reading frame. How about the third possibility of reading frame? The third possibility of reading frame is going to be, now we are GCC, CAA, and GAC. Now, this A and U are not going to account for an amino acid. So we're going to be right here, GCC, CAA, GAC. So alanine, glycine, asparagine. Okay, so these are the three possible open reading frames, right? So whenever we sequence a genetic code, whenever we sequence a genome and we want to know all the different reading frames, we have to actually run these three possibilities. And from there, we are going to say that for a given gene, the first reading frame is the most likely compared to the second and the third. Or the second reading frame is the most likely compared to the first and the third, and so on. At first, it's kind of speculation, and then we actually run experiments to verify that the sequence we thought to be the open reading frame is indeed the open reading frame. But until you run an experiment, you cannot be sure that what you think is a reading frame is indeed a reading frame. All right. So I was saying that the genetic code is universal, which means that all living things evolved from an original living thing. Actually, it is believed that the first form of life that developed on Earth were prokaryotic organisms. All right? And these prokaryotic organisms, bacteria, for instance, read the genetic code the exact same way human cells read the genetic code. And that is the very reason why when you consider an open reading frame in a human gene, you insert that open reading frame in a bacterium, you can produce a human protein in a bacterium. And this is how we produce, we design genetically modified organisms to produce to produce proteins from a human or hormones from a human in a genetically modified organism, such as a sheep, a cow, a pig, right? And so on. Okay? So please remember that. So know that because we have 64 possibilities of codons, right? Some amino acids are going to be encoded by more than one codon. And all codons that encode the same amino acid are what we call synonymous codons. Synonymous because they all encode the same amino acid, right? Makes sense, okay? And then you have codons that are non-synonymous. The non-synonymous are basically that they are producing a different amino acid, right? For instance, let me go to the genetic code right here. Oops, no, not right here. Rather here. So, what are the synonymous codons? CCU, CCC, CCA, and CCG are synonymous codon because they're all responsible for proline. However, CCU and UCG are non-synonymous because CCU produces proline while UCG produces serine. These are non-synonymous codons, okay? All right. So at this point, we are going to take a look at how translation is done. For translation to occur, you are going to need a number of players. You need the messenger RNA that actually carries the information. You need the transfer RNA that actually has an anticodon for an amino acid, right? You have the ribosome that actually does protein synthesis. You need energy in forms of ATP and GTP. And you are going to need a number of protein factors. Now, translation occurs in 3 stages. The first stage is the initiation, the starting of translation. Then you're going to have elongation. Elongation is when amino acids are added one after another, okay? So that you have a nice sequence of polynucleotide, right? And at the end of the open reading frame, there is a stop. That stop is going to allow for a termination, all right? We are going to address that. Now, question, what are where do we start in initiation? How is elongation occurring? And how is termination occurring? All right. So, first and foremost, you have on the messenger RNA, part of the messenger RNA that actually doesn't account for any amino acid, okay, at the beginning of the messenger RNA. That segment is called the ladder sequence. That ladder sequence is essential for the small ribosomal subunit to come and bind. That once that binding occurs, and by the way, this binding is occurring because of hydrogen bonds, okay? Because of hydrogen bonds. When that binding occurs, then you're going to have an initiator transfer RNA that carries a particular amino acid that is called methionine or methionine, depends. And that starting codon is called AUG. It seems that across the living systems, the first codon is 99% of the time AUG, which means that most proteins start with methionine, right? Once you have the complex messenger RNA, small ribosome, small subunit of the ribosome, and the transfer RNA that carries methionine, now the complex, the initiation complex is formed. Now translation can continue with elongation. And that elongation requires the presence of the large ribosomal subunit, right? Because so far we have the small ribosomal subunit that is attached. Now you need a large ribosomal subunit that comes to attach. Okay. So here, the initiation, you have this segment here that doesn't encode for any amino acid. That's the leader sequence. That leader sequence of the messenger RNA is going to attract the small subunit. Through hydrogen bonds, the small subunit will come and attach here to the initiator tRNA that carries methionine is going to bring the anticodon to AUG. That binding is what is going to trigger the attraction of the large ribosomal subunit to come and attach here. And elongation is going to occur. Now that the large subunit has come and bound, what we notice is that in the ribosome, we are going to have now what we call a P site, an A site, and there is a next site that is actually right here is the E site, okay? The P site is the site that carries the growing polypeptide. The A site is the site that is going to accept the transfer RNA that carries the new amino acid to be added to the growing peptide, right? The E site, it's actually the exit site. All right? So now we have the transfer RNA with methionine that is here. Then actually I made a mistake. Let me correct something here. The E site, actually, the E site doesn't belong here. I'm sorry. The E site doesn't belong there. The E site belongs here. Okay? But for now, because we are at the beginning, right, you cannot see that E site. Okay? As the ribosome moves along, the E site is going to come into play. All right. So now we have methionine, we have glycine, then you have the ribozyme that is going to cause an attachment between methionine and glycine, right? Now, that methionine and glycine now connected are going to, they are not, but the ribosome is going to shift this way and keep moving this way. So the ribosome moves this direction. Now, notice that the tRNA that was carrying glycine is the one that is now on the P side. Okay? And now the A side is now empty, right? A new transfer RNA is going to come and sit here, bring the new amino acid. And this is how cysteine is going to come and actually attach right here, right? As cysteine comes and attach, the ribosome is going to slide over, right? As ribosome slides over, now we have the P side that is going to be occupied by the growing polypeptide that is now from 2 to 3, right? And from there, we're going to have another guy that is going to come and attach. And so elongation is going to happen that way, right? So again, we have methionine, glycine, cysteine, lysine, and then arginine, and it will keep growing until the stop codon is reached. And the stop codon is actually a codon that has no equivalent in amino acid. Let me repeat that. The stop codon is a codon that has no equivalent in amino acid. Anytime the ribosome is reading and reaches a point where they cannot add an extra amino acid, you're going to have a dissociation between the ribosome and the messenger RNA, and the polypeptide will be released. But how is this actually specifically happening? So what happens is that at the very end, you have a protein that is called a release factor. That release factor is the one that comes and binds on the stop codon. Again, the stop codon has no equivalent in amino acid, right? So the release factor binds on the stop codon, and that binding is going to trigger two things: a dissociation between the large subunit and the small subunit, that's one. Two, a release of the final polypeptide. All right, now we have a dissociation. Now a translation is completed. That's termination. So the release factor attaching to the stop codon is the one that determines the termination of translation. Now, know that whenever translation is happening, just like transcription, you don't have only one messenger RNA that is produced for transcription. You have multiple messenger RNAs of the same sequence, right? By the same token, when you have messenger RNA that is read, the ribosomes, not only one single ribosome at a time is going to read a messenger RNA. You have multiple ribosomes, okay, that are going to read messenger RNA, so that you are going to have many proteins produced at the same time. All right? So eventually, when the production of the amino acid of the polypeptide is completed, what is going to happen next is that some proteins that we call chaperone proteins are going to come and bind on the final polypeptide. The role of this chaperone is to cause the final polypeptide chain to start adopting a folding. That folding is essential for the function of the protein. Now, proteins can adopt 4 levels of structures. Okay? These 4 levels of structures are the primary structure, the secondary structure, primary structure, the secondary structure, the tertiary structure, and the quaternary structure. But what is the difference between all of this? The difference is that the primary structure, it's nothing but the sequence of amino acid on a polypeptide chain. Okay? That is the initial molecule that is produced when translation is over. Then chaperones are going to come and help the protein adopt more complex folding. And that more complex folding is going to develop because amino acids, some of them are charged, some of them are polar, so that you're going to have some complex interactions that are going to happen within this polypeptide chain. Now, hydrogen bonds are going to cause foldings in 2D shape. Some foldings are called alpha helices. So this is the alpha helix right here. Okay, actually we draw it like this, alpha helix, right? You have Z lines, right? Beta pleated sheets. These are the beta pleated sheets. We also draw it like this. Okay? The alpha helices and the beta pleated sheets, they are all stabilized by hydrogen bonds. In other words, the secondary structure of proteins exists because of hydrogen bonds. I want you to remember that. Okay? The tertiary structure is going to be a more complex folding of the secondary structure. And again here, because of hydrogen bonds, we are going to have more complex folding. So we are going to have some alpha helices here, some beta pleated sheets here, so that we are going to have a protein that is going to develop a 3-dimensional shape, what we're going to call the final conformation. Conformation. That 3D conformation is essential for the function of a protein. Now, let me say this. The tertiary structure is the minimal structural level of protein for a protein to be functional. Let me repeat that. The tertiary structural level of a protein is the minimal structural level that is needed for a protein to be functional. In other words, if you produce an enzyme, that enzyme is in primary structure, all right? That enzyme is good for nothing. It can never catalyze any chemical reaction. Say now, because of hydrogen bonds, that enzyme develops alpha helices and beta pleated sheets. All right? Secondary structural level, well, guess what? That enzyme is good for nothing. Finally, that enzyme undergo further folding and adopts a 3D shape. We are in a tertiary level. Now that enzyme can be active. All right? But here is the last thing I want to say about protein structure. The last thing I want to say is that while the tertiary structure is the minimal structural level for proteins to be functional, however, what we notice is that not all proteins are functional in their tertiary structure. Not because there is an issue with that protein, but because they don't just want to do or don't know how to do a work on their own. So they have to complex with other proteins. As they form complex with other proteins, this is when they are happy, right? If you consider a protein such as hemoglobin that carries oxygen in your system, well, guess what? One molecule of hemoglobin in your blood cells cannot carry oxygen. Two molecules of hemoglobins in 3D shape, perfectly formed, cannot carry oxygen. Three molecules of hemoglobin cannot carry oxygen. You need four molecules of hemoglobin interacting with each other. Only then hemoglobin can carry oxygen the way we know it to carry oxygen in our system. Now, when you have more than one proteins in 3D shape that come together to form a complex functional unit, now we say that the proteins are in a quaternary structure. Okay? So that's a complex of proteins. I want you to remember that. And that quaternary structure is made possible by hydrogen bonds. In other words, after the formation of peptide bonds, and you have a peptide chain, that polypeptide is going to now depend on hydrogen bonds to become functional. All right? I want you to remember that. All right. So the protein folding. Proteins do not fold spontaneously in our system. Now, obviously, you have some level of spontaneity. Yes, that's true. Right? However, the chaperone proteins are the one that play a very important role in causing these proteins to fold properly. Now, but here is another thing that I need to mention. You have two types of proteins that are produced by ribosomes. You have cytosolic proteins, cytosolic proteins. The cytosolic proteins are proteins that are produced to remain in the cytosol. And then you have non-cytosolic proteins, meaning these proteins are produced, but they are not designed to stay in the cytosol. Now, the proteins that stay in the cytosol do not have what we call a signal sequence. Any non-cytosolic protein has what we call a signal sequence. That signal sequence is at the beginning of the polypeptide. Okay? Say we have this as a polypeptide, right? That's the C terminal, that's the beginning, and this is the N terminal, that's the end. Okay? I'm saying some complex things here, but don't worry about it. Okay? So at the beginning right here, you are going to have a sequence of amino acid that we're going to call a signal sequence. That signal sequence is going to determine the fate of that protein, so that as the ribosome is synthesizing the protein and a number of molecules in the cytosol notice the presence of this signal sequence, this is going to trigger the protein that is being synthesized to actually be pulled toward the endoplasmic reticulum. You know that in a cell, you have the smooth endoplasmic reticulum and the rough endoplasmic reticulum, right? And that the rough endoplasmic reticulum looks rough because ribosomes are attached to that endoplasmic reticulum. The question we may ask is, why are these ribosomes attached there? Well, the reason is that these ribosomes actually, as they were synthesizing the protein, the system in the cell noticed that that protein was carrying a signal sequence, and that protein doesn't belong to the cytosol, it belongs somewhere else. That somewhere can be the plasma membrane. You have some proteins that are embedded in the plasma membrane, like this, right? These are transmembrane proteins. These proteins necessarily carries a signal sequence. You have some proteins that are designed to be sent out of the cell. For instance, antibodies are sent out of the cell via a process called secretion. These proteins that are secreted outside of the cell, they have to have a signal sequence. You have some proteins that are part of the lysosomes. They end up inside of the lysosome to make up the digestive enzymes in the lysosomes, right? These proteins carry a signal sequence. Then you have the proteins, while they are part of the membrane, some are part of the plasma membrane, some are part of the nuclear envelope, some are part of the membrane that make up the Golgi apparatus, the membrane that make up the endoplasmic reticulum, and so on and so forth. So you see, the signal sequence on proteins that are being synthesized are very important to determine the fate of where the protein needs to go. But any protein that stays in the cytosol for good never carries a signal sequence. All right? So that's something I wanted to make clear here. All right. So the chaperones are going to play a very important role in protein folding. These chaperones are going to basically allow protein to go from primary structure to secondary structure to tertiary structure and eventually to quaternary structure. Okay? Now, proteins that are misfolded will have to be labeled, because proteins that are misfolded can cause issues in the cell. So rather than letting them be, they are labeled. They are going to be labeled by a protein called ubiquitin. That ubiquitin, one unit is not enough to actually label the molecule to be destroyed. So you need more than one tag. More than one tag of ubiquitin is going to cause the proteasome, which is this huge complex, right, to pull this misfolded protein, stretch that misfolded protein into a machinery, right, where the protein is going to be dismantled, so that we're going to have amino acids that are going to be released one after another. And these amino acids are going to be recycled for protein synthesis, right? Okay, so now not all dismantlement is going to lead directly to amino acid. It can lead to peptides, and peptides are going to be further chopped into amino acid and so on, and that way the system can be actually can recycle the protein. So misfolded protein can be recycled that way. Okay. Now, some misfolded proteins are actually bad news. Okay. They are actually bad news because they cause actually diseases. All right. So this misfolding can actually cause neurodegenerative diseases. Prions are actually proteins that are misfolded. Okay, the conformation is not correct, and because these misfolded proteins can trigger a propagation of misfolding in proteins, this is going to behave like an infectious agent. That's why we call prions infectious particles. And that's why when we talk about microbial particles, we actually mention prions in spite of the fact that they are proteins, right? They cause the issues in our system, severe disorders, right? Say they cause severe disorders and many kind of disorders, right? So the proteasome has to be able to destroy the misfolded protein. If not, these misfolded proteins are going to cluster together, and as they cluster together, they clog our mechanisms, and particularly when it comes to the central nervous system, that can create a significant amount of mess, right? This significant amount of mess is going to be a bunch of neurodegenerative diseases such as the Alzheimer disease, the familial amyotrophic lateral sclerosis. Okay, that is actually okay. Well, let me tell you, in Alzheimer disease, a protein that is called tau protein, the amyloid beta precursor proteins, when they are misfolded, they cause Alzheimer. The familial amyotrophic lateral, this is basically when this is a neurodegenerative disease where the nerves have a hard time controlling muscles, right? This is caused by the misfolding of superoxide dismutase, and superoxide dismutase is an enzyme that is playing a very important role in detoxification of toxic forms of oxygen. But let me close that parenthesis. What I want you to remember, it's that bunch of misfolded protein cause a number of issues in our system. I'm not going to go through each of the issue, but that's what I want you to remember. And finally, but not least, I want to talk about the prion diseases. The prion diseases are subgroups of misfolded proteins, of subgroups of diseases caused by misfolded proteins. Okay? Now, I'm not going to bug you with the history because of the time we don't have, but what we know is that prions actually cause neurodegenerative diseases. And these neurodegenerative diseases are associated with mutations that cause misfolding, and that misfolding, when one protein gets misfolded, the scrappy protein, then it's going to trigger the misfolding of a number of other proteins that clutter the central nervous system and cause neurodegeneration. Okay, so in a human, the Creutzfeldt-Jacobs disease is basically the equivalent of the bovine spongiform encephalopathy, right? Another prion disease is the familiar insomnia and the Gerstmann Straussler disease. I'm not going to bug you with the details of the diseases. What I want you to remember is that the prion diseases are caused by misfolded diseases, by misfolded proteins, I'm sorry. Okay? Prion disease can spread within the body, and they can spread between organisms, right? That's why you cannot actually consume a cow that actually has the mad cow disease and hope to be fine at some point, right? You are going to have issues, right? At some point. It may not be in your lifetime, but if a dog comes and eat you, right? You eat your carcasses, right? They can actually get they can get basically a neurodegenerative disease. We see that in the wild. Okay, that's why I'm taking that example. We see that in the wild. If say a bovine in the wild died of the mad cow form of the disease, right? And say a hyena comes and eat that bovine. Well, that hyena, there is a chance that they can develop a neurodegenerative disease at some point. All right, so let's conclude this chapter here. So you have the scrappy protein, okay, that is misfolded. That scrappy protein that is misfolded is going to target the normal proteins, okay, causing them to change their shape. From there, you have more and more that are going to change their shape. At the end of the day, you have everyone here that is misfolded. And that is a bad news. And this is how prion is going to become an issue. So this is going to conclude this chapter. Thank you for your attention. And we'll talk to you in the next chapter.